Okay, in this video, we're going to look at the other major class of lipids. This is known as the terpenes. Okay, terpenes are molecules that are made up of what we call isoprene units. This is the molecule isoprene here, 2-methyl-1,3-butadiene. Okay, and it's not so much the arrangement of the double bonds that's important as a building block, but it's just the arrangement of the carbons. We just refer to it as an isoprene building block because this is the simplest common hydrocarbon, I guess, that has this skeleton. But don't worry about the double bonds, or don't be looking for the double bonds. It's not a requirement, okay? The carbons can actually have anything on them. They can be C double bond O's, they can have OH groups and various other structures. Okay, all right, so we're going to classify terpenes based on how many isoprene units are making them up. What we're going to find is that the carbon skeletons always have numbers that are divisible by 5, hence because they arise from the 5 carbons of isoprene. Okay, all right. So a monoterpene is going to have 2 isoprene units, and it's going to be 10. A sesquiterpene, okay, the prefix sesqui just means 1.5. It's going to have 3 isoprene units and 15 carbons, and a diterpene is going to have 4 isoprene units and 20 carbons. Okay, so let's look at some representative monoterpenes here. These are all very common plant-derived compounds. Okay, so the simple one here is myrcene. This alcohol that's analogous to myrcene is called geraniol, and this is what gives roses their flavor, their smell. I once bought this compound. I thought it would be nice to have this smell, but after a while you do get kind of sick of it. It becomes quite intense. This molecule here is called carvone. It's a very interesting compound that illustrates the fact that people are full of chiral molecules because the R enantiomer is the essence of spearmint and the S enantiomer is the essence of caraway seeds. Pinene gives pine trees their odor. Okay, and if you look at pine sap, it's very rich in this molecule, pinene. Okay, it'll actually burn because it's got so much of this in there. This is from camphor. It's from eucalyptus tree, and it has this bicyclic structure. And this is the compound that gives Vicks Vapor Rub its odor. Okay, and this is chrysanthemic acid here. It's one of the most potent of all of natural insecticides. This comes from chrysanthemums, and bugs don't attack chrysanthemum plants, and it's because they're quite rich in this natural insecticide. Okay, so let's look for the isoprene units in these terpenes here. Okay, so here's myrcene. Okay, so what we can see here is that we have an obvious isoprene type compound there and one here. Okay, geraniol, same arrangement as myrcene basically of the isoprene units. Okay, and carvone, we can do the isoprene units this way. But that's not the only way we can do it in carvone. We can, let's see, let me change my color here. All right, so I'm going to, okay, pointer options, ink color. We just need a different color. We'll go with blue. Okay, all right, so this is not the only way we can do carvone here. You could have also spotted the isoprene units that way as well. Okay, but it turns out it's not going to make any difference because, for the biosynthetic origin, as we're going to see in a couple of slides. Okay, let me change the Pointer back. Okay, all right. All right, binding is not quite as obvious here because we've got a lot of branching, but we're going to look at actually how this compound is made as well. But we can find an isoprene unit here. In camphor, let's see, isoprene units. Okay, as you can tell, I'm doing this kind of on the fly here. All right, so these carbons here and these carbons here. Again, I'll have these 5 carbons with 4 carbons in a row and a methyl at the middle carbon. Okay, again here and here for chrysanthemic acid. Okay, over here I have a sesquiterpene, isocaryophyllene, and you can see it's a much more complex structure, and I've got the isoprene units sort of color coded in this diagram over here. Okay, let's look at how terpenes are created in nature. They're created from acetyl-SCOA, just like fatty acids are, except there's a different CC bond forming step that gives us the branch point. Okay, so the first few steps are very much like the fatty acid biosynthesis. We're going to have this compound here, which was from the fatty acid biosynthesis pathway. We had the acyl carrier protein instead of coenzyme A, but that's not a big difference because there's a lot of structural commonality in the area where it's bound to the carbonyl group. Okay, so in the fatty acid biosynthesis pathway, we reduce this ketone. Now we're going to add a malonyl SCOA to this, okay? And this is going to give us the branching here, okay? All right, we're going to perform an ester hydrolysis of the ester over here, okay, this ester. Thioester is going to convert to the carboxylate salt. And this is hydroxymethylglutaril CoA, or HMG-CoA, an important abbreviation in biochemistry. Okay, so this is now going to undergo a reduction of this carbonyl group here all the way to the alcohol to provide this. And we're going to phosphorylate this OH group right here. We're going to phosphorylate this OH group. That OH group. Sorry about that. And we're going to now do what we call a decarboxylative elimination. It's going to take out this CO2 group here, and we're going to lose the hydroxide group, and we're going to now have this compound right here, isopentenyl pyrophosphate, which is interconverting with this compound called prenyl or dimethyl allyl pyrophosphate. And these compounds here are the building blocks of terpenes, okay? What happens here is that these couple through a series of carbocation-like reactions and go from 5 carbons to 10 carbons to 15 carbons and get built up 5 carbons at a time. All right, this compound here is called Lipitor. Okay, you've probably seen many commercials for it, and it's what we call an HMG-CoA reductase inhibitor. So chemists at what was then Warner Lambert, now absorbed by Pfizer, said, what if we can make a molecule that looks kind of like the product here, mevinolate, and build into it some other features that will bind the enzyme and not let it go. Okay, so this is actually a very clever designed molecule. Lipitor does not occur in nature. It was designed by the chemist at Werner Lambert. Okay, and what happens here is that because it's got this group here, this actually forms a critical binding to the enzyme called HMG-OCO reductase, and it basically stops it dead in its tracks. So when you take a Lipitor pill, you can't do you can't make these critical 5-carbon building blocks, and as we'll see in just a second, these are critical for making cholesterol. Okay, and so the cholesterol biosynthesis pathway is completely shut down. All right, so let's look at acetyl-SCOA, and let's look at where the methyl group of it goes. Okay, so the methyl group would be there and here in this compound. Okay, so here it is in this compound. Okay. Okay, so we're just going to we'll just do check marks since I can get those carbons in quicker. Or I can make a check mark much faster. Okay. I think I may have added an extra carbon here. Okay, sorry about that. So yeah, this one has an extra carbon in it. All right. And there, and there. All right, so if you look at the isoprene building blocks, what we find here is that the carbons I've shown in blue come from the methyl group of acetyl-SCOA, and the carbons that I've shown in magenta come from the C-double bond O group of acetyl-SCOA. That's going to be important in a problem that we're going to look at in a second. All right, let's just look at some of the types of things that give rise to all these terpenes. So they're really the product of reactions that look very much like carbocation rearrangement. So let's look at nerol and see how it's a potential precursor to limonene and pinene. Okay, so tosylates are too good a leaving groups. So you need leaving groups that would only be reactive in the presence of the suitable enzyme activator. So pyrophosphates fit the bill. They're much poorer leaving groups than halogens or phosphates, but much better leaving groups than acetates or hydroxides. All right. So we're going to take nerol, we're going to convert it to its pyrophosphate, and what we're going to do now is we're going to just make a carbocation here. This is an allylic carbocation, so it would form fairly readily. And it has an alkene here, so we could do an electrophilic attack on the alkene, or the alkene can attack the carbocation to give us a new carbocation here, tertiary. Okay. We could lose the adjacent hydrogen here to make limonene, or we could have this carbon here attack to give us a bicyclic compound that's starting to adopt the structure of pinene. So once you get this carbocation, you could get either alpha or beta pinene, depending on how it eliminates the hydrogen. So it's common reaction, or common terpenes are made through carbocation-like chemistry that you've seen earlier in the class, or in the previous class probably. All right, let's look at a problem here. Let's say this is the compound zingiberine, which gives ginger its flavor. And let's imagine that we're going to feed a ginger plant some C13 labeled acetate. Where's the label going to end up? We're going to look at the problem from the approach of labeling the methyl group of the acetate, or labeling the carbonyl group of the acetate. And we're going to assume that the acetate is going to convert under the biological conditions into acetyl-SCOA. All right, so the first thing we have to do is identify the isoprene units. Okay, and so I've identified them, I've color coded them. So the isoprene units divide like this. This is a sesquiterpene, so there are 3 distinct isoprene units. There are 2 different ways that you can do the last 2. Okay, so I showed one way here and another way like so. Okay, and what you notice here is that no matter which way you go, you're still labeling the same carbons. Okay, so remember, this is 2 slides ago, actually, that in the isoprene skeleton, the methyls and the carbon over here come from the methyl group, and the other 2 carbons come from the carbonyl group. Okay, so when we look at the individual isoprene units, we can assign that if we feed it C13 labeled acetate with a C13 label in the methyl group, C13 label will end up here. And this is a very easy analysis, okay? So remember C13 NMR, natural abundance is only 1%, but if you actually label it, that means the peaks that are labeled become 100 times as large because they're all C13s, okay? So this is a very obvious experiment to do, and this is a very common biosynthesis experiment to determine where carbons originate, okay? All right, so in this next series is where we're going to have the label if we feed it C13 labeled acetate where the carbonyl group is labeled, okay? And what you would see here is that these other carbons are now labeled as a result of this. So we have to find the isoprene units and remember how the biosynthesis is putting the basic isoprene unit together to address a question like this, okay? All right, so what we're going to look at now here is cholesterol biosynthesis, okay? So what we have to do to make cholesterol from terpenes is we have to get 1, 2, 3, 4. So this is 6, I guess this is a triterpene, squalene, okay? And squalene is kind of unique in that this middle linkage here is backwards, okay? Right, so this is what we call a head-to-head. If you look at how the rest of them are arranged, they're arranged to have carbon. Okay, so we'll just label our isoprene unit there. All right, so carbon 4 connects to carbon 1. All right, so if we do the labeling the same way, okay, 4 connects to 4, so that's why we call that a head-to-head linkage. Okay, so that's a opposite linkage. Okay, but there are enzymes that can do that. All right, so under biological conditions, squalene turns into the epoxide, becomes squalene oxide. All right, this gets protonated. Okay. And produces a carbocation. The alkene adds to the carbocation. The next alkene adds to the carbocation again. Now we have 2 rings. This one is going to add, and the molecule folds up into it such a shape that it actually forms the secondary carbocation as opposed to the tertiary carbocation. Here is the same compound again, cyclizing to make the secondary carbocation, which is very quickly intercepted here by the alkene to make another tertiary carbocation. Now we have the steroid skeleton, the skeleton of carbon skeleton of cholesterol. Okay, and how do we now move those methyl groups around? Well, we're going to move them around one at a time. Okay, so we're going to take the carbocation, we're going to do 1,2 hydride shift here to make this. Okay, so this compound here is pictured again. We're going to do another 1,2 hydride shift to make this tertiary carbocation. Now we're going to do a 1,2 alkyl shift. The alkyl group we're shifting being a methyl group, so I'll call it a 1,2 methyl shift. This now generates this carbocation. Another 1,2 methyl shift. Okay, moves this methyl over to here. Okay, and finally, we're going to lose H plus, like so. And this is going to give us a compound called lanosterol, which under biological conditions oxidizes away two of the methyl groups, actually 3 of the methyl groups, and eventually hydrogenates the outer chain, and this is going to give you now cholesterol. So this is how cholesterol is synthesized in nature. Okay, if you shut down the pathway that makes squalene, which is the terpene biosynthesis pathway, which Lipitor does, you stop the synthesis of cholesterol. Cholesterol is actually a starting material for a lot of other molecules that contain the steroid skeleton, so there are a lot of side effects with Lipitor that result from depleting your body of some other good things. All right, let's look at some other molecules that have this cholesterol skeleton, which we're going to refer to as the steroid skeleton. All right, so estradiol is one of the sex hormones, and testosterone is kind of the opposite, but also a sex hormone as well. And let's look at how synthetic compounds derived from these have given us important pharmaceuticals. Okay, so if you just take the alkyne group and add it to this carbon right here, you get ethynyl estradiol, which was the first birth control pill. And it's the compound that's familiarly known as the pill. Okay. Dianabol was created in actually the horse racing industry, I think, and to help when a horse is injured, they want it to rebuild its muscles as fast as possible. So they created an analog of testosterone called Dianabol, which leads to rapid muscle gain. And this is, I guess, many years ago, some football player or something said, if this can help a horse, it can help me too, I guess. So anyway, the idea here is that it was originally created to help you recover from injuries, but it's now abused to build muscles where you already have muscles. Okay, all right, so that's one of, so this has led to steroidal abuse. It's the idea of these drugs started out with a noble purpose, but as you can see, there's the potential for abuse like there is for anything. Hydrocortisone, shown there at the bottom left corner, is very powerful anti-inflammatory. I think it's one of the most powerful ones that if you're suffering from tennis elbow or something like that, you'll get an injection of cortisone, and that typically relieves the problem almost instantly. Okay, the last two compounds, tamoxifen and diethylstilbestrol, are not steroids at all, but when an enzyme that uses steroids sees these molecules, it looks like one to them. So tamoxifen is actually an important treatment for recurrent breast cancer, and it's actually one of the most important treatments. And if you look at, let's see, so parts of it look kind of like a steroid. Okay, so this part here looks kind of steroid-like, and it inhibits an important biological process that involved in breast cancer recurrence, and it's a very important treatment for that. Okay, diethylstilbestrol is a molecule that it reduces miscarriages, but it has a lot of side effects. It's called DES. It's still used in the agricultural industry, but not in the not in humans anymore. It has not. Let's see. I don't remember the side effects of diethylstilbestrol. It's not like thalidomide or anything like that, but it's still undesirable, so it's not used in humans anymore. But you can see that it sort of, again, if you let's just put this methyl this way instead of this way, and put this methyl this way instead of this way. And when you look at it, you can see that it's starting to a biological entity, it's starting to look like estradiol to some extent. It has the two hydroxide groups in the right place. Okay, all right, so that's it. If you looked at the book, you probably noticed that this chapter is a lot longer than what I've talked about, and I'd like to think of this chapter as a prelude to biochemistry or medicinal chemistry, and you can take more advanced courses if you want to see this chapter expanded a little bit. Okay, so that's where this one is going to end for now. Okay.